Alright, so I want to talk about the bacteria growth cycle. Okay, and the bacteria growth, or in many cases, this is called the bacterial growth curve. It has basically four phases, and those phases are a lag phase, a log or exponential phase, a stationary phase, and a death phase. Okay, so the lag phase is when the cells are transferred to a fresh medium. So you have some cells in an older culture, let's say, or on a solid culture, transferred to a new, fresh medium or media. They require time basically to detect the environment, and during the time that they're detecting the environment, they begin to express specific genes and synthesize components necessary for rapid growth. Okay, and the cells are not dividing at this time. That's why you see this lag phase. That's why it's called a lag phase. At this point, there's no division, so there's no increase in the cell number. Okay, you just basically preparing, growing, increasing in size, getting ready to divide. The next phase that they enter is basically this log phase or exponential phase, okay? And in this phase, the binary fission is occurring at its maximum rate. So binary fission is the way that bacteria divide and produce new cells. And this is occurring at maximum rate, and that's why you have exponential growth. So they're basically dividing as rapidly as possible and producing a large increase in the number of cells. So here you wind up with a net increase in the number of cells, okay? Net increase in the number of cells. Then it enters what arguably is the most important stage, I would say, in the bacterial growth cycle, and that is the stationary phase. So at this point, the growth is stopped, so there's no net increase, okay? There's no net increase or decrease in the number of cells. So there's new cell, basically the same number of cells are dividing as are being killed. And at this point, the bacteria use new forms of metabolism. This is where it becomes important for human use, is the bacteria use new forms of metabolism to survive, okay? And in some cases, they're producing secondary metabolites, which are often useful to humans. In some cases, this is where the antibiotics are produced, okay, or antibiotic molecules are being produced. And those then can be isolated. So it might be important for me to, and they call this a chemostat. It basically keeps the bacteria in a stationary phase so that they can continue to produce whatever that secondary metabolite that is useful to humans is, and it can be extracted then and used for that purpose. And also at this point, spores are formed. Okay, and the stationary phase is where you might find the formation of endospores. Okay, and endospores are usually produced in cases where nutrition is lacking, the nutrients are lacking and beginning to become scarce, and of course there's a large number of cells, so that means they all require a high level of nutrition. And in some cases, in certain bacteria, spores are formed, and those spores are really, really resistant to both drying out and heat, and they can actually wind up surviving a very, very long time basically in a dormant state. And the final stage is this death phase, okay? And at this point, more cells are dying than are dividing. And what ends up happening here is that there's a net decrease in the number of cells, okay? Less cells dividing, more cells dying, results in a net decrease in the number of cells. So that's basically what's going on here with the curve. Now, I might want to just say a few more things about the stationary phase. So basically the cell number stops rising at some point, okay? During this log phase, this is eventually going to level out. And it usually levels out as a result of a buildup of waste products or just a lack of key nutrients. And at this point, the growth curve, levels off. The culture is in what's called the stationary phase. And like I said before, the stationary phase can be really important. If you contrast this with some eukaryotic microorganisms, such as protozoa, they enter the stationary phase at a much lower cell number. Okay, so they cannot tolerate the same cell numbers as the prokaryotic cells, but nonetheless, it's very similar. And because the cells are in stationary phase, they are not metabolically nimble as the cells in the exponential phase, okay? So they basically don't have that same ability. And they could also be being damaged by oxygen radicals and toxic waste products that are a result of the metabolism, and rapidly do kill microorganisms. So of course the stationary phase is also kind of a dangerous time. That's why you don't see the increase in the cell number. So as an avoidance strategy, some bacteria differentiate into very resistant spores in response to nutrient depletion, while other bacteria undergo less dramatic but effective molecular reprogramming. Okay, and that molecular reprogramming is what I call, the production of secondary metabolites, and those metabolites being very, very useful for in some cases for humans. So setting up a continuous culture, and I kind of hinted at this, that there's something called the continuous culture, which maintains constant cell mass during exponential growth. Okay, it maintains constant cell mass rather. So in an open system where fresh media is continuously added and old media is pulled out, the equal amounts of old media are siphoned off. Bacterial populations can be, of course, maintained in specific growth phases. All right, so if I want to maintain them in the exponential phase, I can do that. If I want to maintain them in the stationary phase, I can do that. And that is a result of just, adding fresh media and removing old media and old cells, okay, and dead cells and things of that nature. In this type of growth pattern, this is known as a continuous culture. And all cells in the population achieve what's known as steady state. And they can be maintained for a very long time. And I said that there's a specific type of machine or a specific type of culture setup that you can go through. It's known as a chemostat, and it's basically a continuous culture system in which, again, a limiting amount of essential nutrients are added, and some cells and old media is removed. And this is held in specific ratios and specific concentrations in order to achieve the proper phase. So either the exponential phase or stationary phase, wherever I want to keep that culture.